Our protocol provides a simple method for 3D cell culture and is highly amenable to downstream analysis. This facilitates the evaluation of heterogeneous patient samples and their specific drug responses. The advantage of this technique is that spheroids can be formed from small cell numbers, allowing for both preservation of scarce primary samples and high throughput screening for patient-specific drug responses.
The hanging drop model creates a physiological environment with 3D drug diffusion and dose responses corresponding to tumor stage. This allows for the development of targeted therapies and patient-specific treatments. This method is ideal for studying ovarian cancer heterogeneity and the development of chemoresistance. However, it can also be used to study other cancers and drug-resistant cell populations. When plating spheroids, it is important to pipette precise volumes. This ensures consistency between spheroids. The spheroids and their respective droplets are inherently fragile and easily lost without proper handling. Therefore, we will show how to appropriately plate, maintain, and analyze a hanging drop plate. Demonstrating this procedure will be Michael Bregenzer, a graduate student from our laboratory.
Start by filling each well of a 6-well plate with 4 to 5 milliliters of autoclave deionized water and sandwiching the hanging drop plate between the lid and the bottom of the plate. Add 800 to 1,000 microliters of water around the rim of the hanging drop plate to provide a humid environment and minimize evaporation. Next, collect 2D grown cells by detaching them with trypsin and then adding 6 or 8 milliliters of medium containing FBS. Aspirate the cells with a 10 milliliter